Good afternoon, students. With yesterday's completion of fertilization, I have to now deal with the next part, which is called sex determination. A very short note is given for generalization of the concept, like whether male spermatozoa is responsible for determining the gender of the offspring, or it is the female gamete, the egg or ovum. So, if you could have a small revision, yesterday you said in fertilization, haploid male gamete fuses with the haploid female gamete, secondary oocyte, ovum, to result in the formation of a diploid structure called as zygote. So events were like this. First, we focused on ovulation. We do have to mention it because without ovulation, else there is no way or meaning for fertilization process itself. So the female gamete on the 14th day, LH surge due to activity of LH hormone by the pituitary. Even though both FSH, LH are in peak level, LH surge will occur rapidly. So Graafian follicle is pushed to the surface of the ovary. One dominant follicle ruptures, oocyte is released. Oocyte will reach the body cavity coelom. Fimbria picks up cilia activity of fallopian tube, and even the muscles of the fallopian tube will take the oocyte into the region called ampulla. Right? So that follows insemination process. Second, it was insemination. In insemination, you said male during copulation ejaculates nearly 200 to 300 million spermatozoa into the vaginal tract. So that process is called as insemination, has to occur after insemination when the spermatozoa, the semen, is released in the female reproductive tract. Therefore, the word internal fertilization, right? In the vaginal tract, it is been released. Third very important thing is that these sperms should undergo capacitation, very essential. In this capacitation, what did you say? The sperm plasma membrane, whatever is in the head region, decapacitizing proteins should shed off. They should go off efflux by taking in of calcium ions. So calcium ions move into the head, and decapacitizing proteins are thrown out of this sperm region. So this makes the sperm to be active. Not only active, sperm gets the ability to fertilize only if undergoes capacitation. Okay, so if capacitation occurs only, sperm is able to penetrate the oocyte. So removal of these proteins occurred by influx of calcium, correct? So this took place where means it took place in the uterus, it took place in the fallopian tube. Number of duration, like time taken for this capacitation was 5 to 7 hours as mentioned in one of the books. After capacitation, the sperm reaches the oviduct, that is the ampulla region. The chemical attraction between the sperm and ova, that's otherwise the sperm will never move towards the ova. Ovum or secondary oocyte is releasing the fertilizing chemical by its membrane, while sperm releases anti-fertilizing. So fertilizing, anti-fertilizing chemical attraction makes both the journey to come together. Okay, then sperm's head has acrosome. Right now, next process is number 4, acrosomal reactions. You said the acrosome burst, releasing the lysins called as sperm lysins. So they are made up of hyaluronidase who dissolves corona radiata, zona lysins who dissolve the zona pellucida. So हमारे एक के जो बाहर membrane थे, that is what you call secondary membranes. Corona radiata and zona pellucida both have to be like digested. That is done by the acrosomal reactions. So after breaking that, the point when the sperm touches the egg membrane, that is primary membrane, we call that region as fertilization cone. Egg now, who was polarized, now it is depolarized. The egg is now in depolarized state. So finally, the sperm head, sperm ka middle piece, rarely tail. Okay, so energy is available by mitochondria. The sperm makes the entry. So next, after acrosomal reactions, what is that? The sperm is going to make the entry into the secondary oocyte. Now what happens when the sperm enters? Very important things are going to occur. What happens means the polar body, right, will be released out, and now the secondary oocyte is called as ovoid. The centriole will be contributed by the sperm. Sperm contributes the centriole. So second meiosis will be completed in the oocyte, releasing one more polar body, and now this structure is called as ovum or ovoid. That's very fine. Next, after this, immediately cortical reactions occur. So what is this cortical reactions means? जो भी आप plasma membrane के नीचे cortical granules थे, right? So that will be extruded outside with the help of calcium ions to make a very thick barrier. What do you call that layer as? Hyaline layer or zona pellucida gel layer. Why did you make the zona layer very thick? Means just to prevent the polyspermy, means no more sperm or second sperm should not enter inside. That was our attribution. So that was the reason for preventing the polyspermy. The zona layer becomes thick. This is called cortical reactions. After the sperm enters, cortical reactions will happen. So second sperm entry is inhibited or prohibited. So ये बहुत ही अच्छा हुआ. Last, after the sperm entered, now the male nucleus, sperm में जो nucleus था, it swells, called pronucleus. The female egg is activated. The female nucleus also swell. It is called female pronucleus. दोनों भी nucleus membranes will fuse together and both the chromosomes, paternal chromosome and maternal chromosome, will mix. 23 plus 23 haploid. Now it is a 46 chromosome celled, one celled condition, zygote. One cell, now it is having how many chromosomes? 46 chromosomes. So that is what we call it as zygote. So this is a short flashback of the yesterday's class called fertilization events. Now I have to talk what is this sex determination. Very small concept is given in your book. That follows sex determination. So what is in this sex determination? It's very simple. Here a male person and here it is female person. So we marked it as to be a female. So male and a female. Male makes chromosomes, sperm, with autosomes plus X chromosome and autosomes plus Y chromosomes. Right? He is heterogametic. The person is heterogametic. The male makes two types of sperms, 22 plus X, 22 plus Y. So he is heterogametic. While female makes the oocyte, ovoid, whatever you call, it is only one which having only X chromosome. So we call it as homogametic. Homogametic condition this is. So this is the fact of yesterday's fertilization. Further, one note is given. Now, because he is heterogametic and she is homogametic, the egg will be having only 22 autosomes plus X chromosomes. So this is 22 plus X or 22 plus Y. Now question is chance factor. It is a chance factor. It is not intruded, it is not voluntarily, or it is not planned also. It is a chance. What chance means? If this sperm with the X meets this oocyte, which is called as X, the resultant zygote will be X and X. So that is going to be a female. That's very simple matter, every one of us know, but still. Next, if this spermatozoa among the millions of sperms, even by chance, if this happens to meet this, which is called as X chromosome, right, of the female ovum, if this sperms meet this ovum, now that resulted in the form of XY chromosomes in the zygote, and that zygote shall be declared as a male zygote. Hence, what's the conclusion for sex determination? In the society, even though we have blames on a female, science clearly depicts that male is responsible for, right? Male is responsible for gender determination. That's it. Male is responsible for gender determination. If male is responsible for gender determination. So this is the case of sex determination. A small concept, enough. It is absolutely easy to easiest. XX and X chromosome here, X and Y. XX female, Y and X, XY is male. Next. Embryonic development and pregnancy. Let us see what context has to be added for today's class in embryonic development and pregnancy here. Now, continued the part next from this site, which is already a zygote now. Embryonic development, what further classes? Here we are in the region which is called as ampulla. Now, soon after fertilization, soon after the process of fertilization, next what? The 2n single-celled zygote. 2n single-celled zygote, right? So it's diploid zygote with single cell. Yes, so this is now single-celled structure, zygote. This begins cleavage. Zygote begins cleavage. Now, what do you mean by cleavage? Mitotic division of the zygote resulting in the formation of embryo. What do you mean by the word cleavage? Now, what does the cleavage occurs? Watch here. This is same size. Zygote remains to be same. The cell of the zygote will be equal to cell of the morula also up to 16-celled stage. The cell of the zygote, size of the zygote shall be same. Number of cells may be more, right? Now, cleavage begins like this. One cell divides and make two daughter cells. Absolutely like this, two daughter cells. Are to maintain the same structure further, right? Embryonic development. All those events occurring that leads to pregnancy. 2 divides into how many cells? 4 cells. 2 cells divided into 4 number of cells. I am making very simplified diagram. Now let me pause here at this case. So internally 2 will split and make 4 cells. But if this, in this case, the splitting of the 2 cells is in such a way that if both the cells separate out and make 2 zygotes, right? So division is so rapid that each of these cells split individually into separate cells. Now what is this? This is also one cell zygote. This is also one cell zygote who is independent and separated, but both are product of one cell, one ovatid, one sperm. But here split into 2 individual cells. Both will start again first cleavage once again. So you will again make 2 cells. 2 will make 4 cells, right? One cell into 2, 2 cell into 4, 4 into 8. You shall also 1 into 2, 2 into 4 cells, 4 into 8 cells. So under this pattern, 1, 2, 2, 4 cells, 4 into 8 cells. But if in this first cleavage, under 2-celled condition, the split was so rapid that both the cells separated out, then you can expect this to be as identical twins. So these 2 kids will be identical twins, also called as maternal twins, because both the twins are a product of one spermatozoa and one ovatid. Hence, they both will absolutely look 100% same genome, 100% same body morphological features. So this results in the form of maternal twins, which we call it as identical twins. Right? Maternal or identical, having same genome. Well, enough, that was an information. Continue next. One single cell into 2 cell, 2 cell into 4 cell, 4 cell next into what do you call? I should write it as 8 cells. Please take care that the size of the cell should be same. Into 8 cells. After 8 cells, I will go up to this cell which is called as 16. So this number of cells here could be 16. Here let me make it as 8. Let me make it as 8 cells, and now this is 16. Now question, what do you call for each of these cells as here? Each of the cell is called as blastomere. Blastomere, cell of the zygote. Cell of the zygote who now developing into embryo. Zygote started cleavage. What is cleavage? Mitotic division of the zygote to develop into embryo is called as cleavage. Each cell of the embryo is called blastomeres, right? So let me write here as further development. I can't keep this zygote here. Zygote will be dividing into 2-cell stage, 2-cell stage followed by a 4-cell stage, 4-cell stage followed by 8-cell stage, followed up to here what you call a 16-celled stage. When you reach the 16-celled stage, you are towards the end of this part of the fallopian tube, and by the time here 16, which moves down to the uterus, at the 32 you are going to attach to the wall of this endometrium, and I call that process as implantation. So there is some other information there I will tell, but here it started from 1 cell to 4, 6, 16, comes down. By 32, you are ready to get attached to the wall of the endometrium. So I call this process as implantation. So let me complete this word. What is cleavage? This is called cleavage. Cleavage is nothing but mitotic division of the zygote. Mitotic division of the zygote leading to the formation of embryo. Leading to the formation of embryo. Mitotic division of the zygote leading to the formation of embryo. So that is about our word what is called as cleavage. Each cell of the embryo is called as, each cell is called as blastome here. Now, 8 to 16 celled stage, right? 8 to 16 celled stage, whatever is that 8 to 16 celled stage is called as morula. The embryo is now called as morula when it is in 8 to 16 celled stage. It looks like a ball of mulberry cells. Absolutely, it looks like a ball of mulberry fruit. Absolutely, if you hold, it's very small red color fruit called mulberry fruit. So this mulberry fruit is resembling our morula, who is 8 to 16 cell stage. The embryo is called as morula. Further, next. After 16 cell stage, now the embryo is distinguished into two layers. After 16, till then it's not shown or not discussed. After 16 cell stage, you are being derived into two, distinguished into two layers, right? The embryo after 16 celled is distinguished into two layers, right? Outer trophoblast. It forms outer trophoblast or trophoectoderm, trophoectoderm or trophectoderm, inner embryonic cells. Outer trophoblast, inner embryonic cell mass. Now how does it look? Further, after 16 celled stage, embryo derived into two layers of tissue, outer trophoblast. Watch this. Outer layer called as trophoblast. Outer layer called as trophoblast. Correct? Now, this is called as embryonic cell mass. This is called as embryonic cell mass. Yes. So this is, let me color up this. Outer layer called as trophoblastic cells. Further embryo I am talking after 16 cell stage. This is what we see. And this is outer trophoblast cell. This is inner embryonic cell mass. Embryonic cell mass. And you develop a cavity because of rearrangement of these cells. A cavity or a space is arranged, observed because of rearrange of these two types of cells. Smaller trophoblast, larger embryonic cells. So this cavity is observed, known as blastocele. Cavity called as blastocele is observed. Such a cell with two layers. Now at this condition, outer trophoblast, inner embryonic mass with the cavity called as blastocele. This structure of the embryo is called as blastocyst. This stage is called blastocyst stage. After morula, next stage is called blastocyst. If you could tell, sir, number of cells within that embryo is 32 means, yes, I will tell, because morula is 8 to 16. Usse zyada blastocyst, yes, you can dedicate that to over here. Now comes, what does this blastocyst to do? Sir, ये होने में कितना time लिए आप, right? ये करने में means from here to up to 32, which is I'm talking blastocyst. It has been now 1 week after fertilization. And this next thing going to happen is implantation occurs. Who undergoes implantation? Morula undergoes implantation? No. Blastocyst undergo implantation? Yes. So blastocyst होने में कितना time लिए? 1 week after fertilization, right? 1 cell into 2 cell, 2 cell into 4 cell, 4 into 8, 8 into 16, 16 into 32. This was not a matter of hours, it was a matter of 1 whole week after fertilization. So next what I am going to do is, After 1 week of fertilization, after 1 week of fertilization, after 1 week of fertilization, blastocysts undergo implantation. Blastocysts undergo implantation within the endometrium of the uterus. Within the endometrium of uterus. Blastocysts undergo implantation. Now, where does implantation occur? How does implantation occur? So, let me zoom up this. This is what called as implantation. After 1 week of fertilization, right, blastocyst undergoes implantation within the endometrium of uterus. Now watch this. This is the, let me take uterus. This is the inner layer of the uterus called endometrium. This is perimetrium. This is middle myometrium. Hope this is innermost endometrium. Okay, so this same picture is taken here. That's it. The same picture is elaborated. Watch. Outer trophoblastic layer of cells. Outer trophoblastic layer of cells. They elongate, they stretch out and they elongate, and they make invaginations within the endometrium. I call this tissue as chorionic villi. This is called chorionic villi. What? Trophoblast cells, they make invaginations, elongated cells. Invagination means stretched, elongated cells. They will stretch out, they elongate and make invaginate into the endometrium, and this structure which is newly developed is called as chorionic villi. This is of fetus. I am talking about what is this outer trophoblast doing. What does this inner embryomas? Still not yet said. Outer trophoblast layer did this job. Now what does maternal tissue does? Maternal tissue will also make interdigitations like this. They also make along with that, exactly, if chorionic villi comes up like this, mother's tissue will also fix in between these tissues extensively. Mother's tissue will also develop in between these two, and now this is tightly fixed between the two interdigitations, so that certain things can be exchanged between the two. So this will be now developing into a new structure called placenta. This is what now going to become structure called placenta. So this is trophoblastic cells, mother's tissue, both will become interdigitations. This is a structural functional unit of the fetus now, from the mother to the fetus, for exchange of that, these, etc. That is now called as placenta. Okay, so let me write. So further, I'll erase this. We are talking about embryonic development only now. Now, maternal tissues plus blood vessels responsible for making interdigitations also show interdigitations. Maternal tissues will also show interdigitations with chorionic villi. With chorionic villi, and become structural and functional unit of the fetus development. It's not of mother's contribution to the fetal development. So structural and functional unit of the fetal development, and that word is now called as placenta. Right? Yes. So we wanted to get the word, what is this? Placenta. Soon the entire, this tissue, mother's tissue, not only up to this region, mother's tissue will start growing across this. Mother's tissue starts spreading across this entire trophoblast cells, means this what you call blastocyst cells. It starts growing and completely takes in this blastocyst into the uterus endometrium. Right? It will completely grow. Don't worry. This mother's tissue will completely grow, surround this embryo and take into the wall of this endometrium, and that will be the beautiful word called as implantation. So that is completed the word implantation with the formation of the word called as placenta, right? Now these tissues will be developing into placenta. This is trophoblast cells also. Kindly remember, trophoblastic cells are here. Surrounded by that, mother's tissue is also here. So mother's tissue surrounds the blastocyst, embedded into this wall called as endometrium, and now this is declared as implantation. So question is, when does implantation occurs? Means after 1 week after the fertilization occurs. Both the tissues, what? Chorionic villi plus maternal tissue plus blood vessels, they interdigitate themselves among themselves, and they develop into a functional structure called as placenta. Okay, so that makes the word what is placenta and what is this next called as implantation. Now continue next. What is this proper embryonic cells do now? Next part. Yes. Inner cell mass. So what does inner cell mass do? Inner cell mass, they rapidly divide. Inner cell mass rapidly divide. They rapidly divide and differentiate into 3 layers. And differentiate into 3 layers here. ये बाहर का जो भी है, that is fine. It's already becoming placenta. Inside, just watch. Inner layer of embryonic cell mass like this. Yes, 3 layers. Inner cell mass differentiate and rapidly divide and make into 3 layers. Number 1, outer ectoderm. Next, middle mesoderm. Embryonic cell mass, I am telling, inner cell mass of the embryo. Inner cell mass of the embryo, it makes embryo proper. It makes embryo proper. Inner cell mass only is responsible for development of the individual or adult. Right? So with these 3 layers only, we are developing into a fetus, into an adult. It has all the necessary information to develop into an adult. Outer ectoderm, middle mesoderm, and innermost endoderm. And innermost endoderm. So now, what is this innermost layer speciality is? Innermost layer of the speciality, we call it as even stem cells also. The innermost layer is called as stem cells. They can be preserved for future use, for developing any other organs in case if it is needed. Inner layer of the cells called endoderm, they are also known as stem cells. Innermost layer cells are called as stem cells that can be preserved also, because each cell has the layer to develop into an respective organ. In future, if any of your liver or kidney or something is damaged, these stem cells can be grown and made to develop into a respective organ in laboratory condition also. That ability is there for this innermost layer of the cells, which is called as stem cells. Remember the word stem cells. So I need not find out where it is. Innermost layer means it is understood this layer which is concerned to endodermic layers. That's all. Over. So this is the story of these layers, which is called as embryonic cell layer. So this was the story of number 1 layer, which is called as outer trophoblast. So now this completed the further embryonic development. Now next is we have to go for pregnancy, that is called gestation, and the organ which is called as placenta. Right, now what does this placenta do and how much will be the gestation period? So we will watch that next. Okay, in the further process of now pregnancy. So what next to be dealt? Before this diagram, there is one more word. During implantation, what makes implantation to occur? One other layer is also there. Here, primary plasma membrane. This is highly sticky. Just a minute. Very essential information. This is highly sticky. Other than this, you have the layer outside to this vitelline membrane. And now this was zona pellucida. This was zona pellucida. Outer layer which is called as corona radiata cells. Outer corona radiata cells. Plasma membrane, highly sticky membrane. Zona pellucida layer is there. So implantation will not occur unless zona pellucida layer is dissolved. That is very most important. So zona pellucida layer has to be dissolved. Right? Zona pellucida, which is a thick layer, gel-like layer. This zona pellucida has to be dissolved. Once only this zona pellucida means outer to that corona radiata is also there. They both should be completely dissolved. Then only you are going to expose this plasma membrane, who is highly sticky. Then only this, whatever the layer of cells, what you call trophoblast, is able to make the process called as implantation. Okay? In future, that embryo cells, जो भी आप बोले थे, trophoblast cell and inner mass cells, before that, outer zona pellucida layer has to get dissolved, exposing the highly sticky plasma membrane to get attached for the implantation. So that word kindly note. Right? It's essential part of your textbook only. Now, going for the next part, pregnancy. Before that, what is the function of this placenta now? What is the function of the placenta? Placenta and its functions. Placenta and its functions. So, what is placenta? Structural and functional unit of the fetus, whose mother's tissues, mother's blood are in direct contact with that. And placenta के जो भी function है, it is not directly with the fetus. The placenta function will be connected with the help of umbilical cord. Whatever this placenta is doing its function, placenta is not in direct contact with the fetus. Placenta is in not direct contact with the fetus. The function of the placenta is brought to the fetus with the help of this structure coming down from the placenta known as umbilical cord. So you call it in local word, hokkala balli. What do you call? Umbilical cord here. Yes. So that brings all the necessary development for the fetus. Now coming to this, functions of placenta. Placenta help in transport of oxygen, help in transport of nutrients. Of course, no. Right? It acts as transport oxygen, transport nutrients. Placenta collects wastes of the fetus. It collects waste of the fetus means it acts like a fetal what? Kidney, right? Which is equal to the fetal kidney. Fetal kidney. Placenta stores food. That is transporter supplier. Means it acts as a fetal liver. It can act as a fetal liver. So here you can see fetus ka liver abhi nahi functional, not started, but it can store the food in the form of a minute structure marked here as yolk plug, right? Marked here as the yolk plug. That could be even because of the extra embryonic membranes. Along with that, food reserved to be stored for the fetus because liver is not yet developed. That is not marked to be as a yolk plug, right? It is of the placenta's function only, developing into yolk plug. Next, collects waste. Placenta collects carbon dioxide. Transport oxygen, nutrients. Nutrients means food, right? So it is understood as food. Essential food it transports. Remove the waste, stores the food, collects the carbon dioxide. Placenta, very essential, transport antibodies. Placenta transport antibodies. Passive immunity for the fetus. Immunity for the fetus, passive immunity, right? So what is this passive immunity, that is in the form of immunoglobulin G. So IgG antibodies can cross placenta. So antibodies are passed through the placenta, mother to child, fetus, passive immunity is also given. Now, most essential than this, placenta makes hormones. Why jabardast hai? Hormones what are produced by the placenta, what do, right? So that comes under functions of placenta only, but hormones role is very authenticated. Now continue next. Placenta functions. Placenta also secretes. Placenta also secretes hormones. Also secretes hormones. Which are those hormones? Watch. If I could wish to give you additional information, if it is beyond, right? So at the age of, so around 2 weeks of pregnancy, not age of, excuse me, at around 2 weeks of pregnancy, placenta first releases a hormone called HCG, human chorionic gonadotrophins. Human chorionic gonadotrophins. Human chorionic gonadotrophin. That is marked immediately after 2 weeks of pregnancy. That is like after 2 weeks of fertilization. Right? 2 weeks after fertilization, after implantation only, just within the span of 2 weeks after fertilization. You see, human chorionic gonadotrophin, HCG. What does this HCG does? Immediately, this HCG will act on, it will act on corpus luteum. Corpus luteum. It will act on corpus luteum, making the corpus luteum at around something called as 3 weeks. After around 3 weeks, right, to secrete larger amount of hormones called progesterone, right? Corpus luteum to release a hormone called progesterones. It maintains corpus luteum, it acts on corpus luteum, stabilizes corpus luteum, right, by around 3 weeks up to a month of, say, 3 months of pregnancy, right? Conception at the, what do you call, first 3 months, the embryo is very, very sensitive, right? For any of the infections of bacteria, for any infections of virus, there is a more chance of misconceptions, right? Miscarriages, abortions ho sakte hai up to 3 months. So progesterone have major role in maintaining the pregnancy, thickening of the endometrium, right? 3 weeks se 3 months, like more secretion of hormone called as progesterones are going to be released by corpus luteum. Not only that, you also release, corpus luteum also release a hormone called as estrogens. Estrogens. They maintain the female characters. Menstrual cycle will be stopped, right? So the whole menstrual cycle is arrested. FSH, LH is absolutely ceased. So which are the prime hormones coming down during this from the placenta? Placenta function, they also secrete hormones. HCG, correct. Progesterone, correct. So I will circle up the hormones. HCG, progesterones, estrogens. Other hormones released by the placenta is HPL, right? Not IPL. IPL match nahi hai, ye HPL hai, human placental lactogen. Human placental lactogen. And at last, a hormone called as relaxin. During the what process, right? In that whole pregnancy period, we will be expecting relaxin. But placenta makes only these many hormones, only 4, because relaxin is released by the ovary. Let me make that also clear. Relaxin is released by the ovary. So don't get add with this placenta. Relaxin is produced by the ovary. HPL, placenta, estrogen, placenta, again progesterone also placenta, plus corpus luteum. Corpus luteum releases progesterone, estrogens. So total 4 hormones released by your placenta, HCG, HPL, estrogens and progesterone also. But corpus luteum also releases larger amount of progesterone and also releases the estrogens. So which are the hormones released only during pregnancy? That's also a question. Only during pregnancy means only during pregnancy. Which are the hormones only during pregnancy means HCG, HPL and relaxin. This much only is to be answered. That is only during pregnancy. So which are the placental hormones means 4 hormones. Which is the ovarian hormone means ovary will release one more hormone. That is only during the pregnancy I am talking. That is called as relaxin. Plus, not only this, I have to talk about other hormones also. वो दूसरे कौन से hormones है who will also take part during this pregnancy development means it is progesterones, estrogens, prolactins, cortisols and thyroxines. These many group of hormones will also increase, these hormones will also be increased several folds. Sikapate, too much of hormones will be released. These hormones will also be released during the pregnancy, correct? So, placenta makes those hormones in her body. These many hormones are also released several folds. Just why means due to increased metabolism of the mother and to increase the fetal growth. That's our intention. Just to increase the metabolic activity of the mother and to increase the fetal growth. Now, because of these many hormones and your other hormones of pituitary plus in your stomach gastrin hormones, maybe hormonal upside down, the person who the female, because after conception, this what you call second week or third week is going on, HPL, HCG and all are released, she happens to get nausea, vomiting. So vomiting and you call good news, congratulations, right? So that the matter you could call, sir, wo aurat kyun vomit kyun kiye? If you want to ask, why does the female gets the nausea and vomiting? Yes, and you could call her something, good news is there. Yes, why good news? Good news is here. The reason, because of these increased hormones, the other hormones of your gastro tract may be suppressed and nausea, indigestion, abnormal things could occur. Metabolic activity in the body is high because for fetal growth here in these 2 or 3 weeks, nausea is because of these hormonal imbalances. That's all for a short duration. It could go for another 1 or 2 months. Up to 3 months, these things could happen. So the fetus is very sensitive up to 3 months. That is first trimester. So we'll see again next class. It will be continued. So what could happen in the different stages of pregnancy, 1 month, 2 months, and what is the gestation period. After gestation, parturition occurs, followed by the lactation occurs. So today you just completed embryonic development, you started, then you spoke about the placenta, development of placenta, functions of placenta, hormones of placenta. So what is the duty of relaxin? We will tell again in the next classes. Your pubis symphysis के ligaments को relax करेंगे. Contraction of smooth muscles, no, that is oxytocin. So relaxin function is different, oxytocin function is different. So दोनों को please don't make together. So relaxin, what do you do? Relaxes the pubis symphysis ligament. So widen the pelvis for the process called as labor birth. So we shall again meet tomorrow evening. Yes, tomorrow is there. We will again come and continue in the next class. So I will stop here right now.